In this video, we want to talk about our eye optical system. So before starting, I need to explain that, in this video, we don't want to talk about the biology of our eye. We just want to look at it from pure perspective of optical system. We want to look at it as like a camera system or a lens system that, you have CCD, you have different surfaces, and the end goal that in future I'm gonna make another video that we can import this model into Zemax and look at it like a pure optical system. So eye is a very complicated system. It has veins, water, nutrition, lot of things. But in this video, we just want to talk about very simplified optical system. Even the optical model is not that complicated. I choose one of the simplest, one of a very simple optical system for this video. So let's start and talk about the main components of the eye. The first component of our eye that interacts with outside of our eye or the light is cornea, the first surface of our eye that has two surfaces, and the refractive index we can consider it as a constant refractive index. The second one is anterior chamber, which, as you can guess from the name, is like a chamber that there is something in between, and that material is like a jelly type material. The refractive is pretty close to water, and it keeps the shape of the cornea like spherical shape. The third optical element is the pupil. That is very important for our optical system. It acts like a stop of our system, or like an iris that you can change the size. It's not a constant diameter. I'm gonna explain that the diameter of this part of our eye is changing because of the intensity of light. The next one is the lens or crystalline lens of our eye. This part of our eye is a kind of adjustable lens that you can change the focus point of this lens, and we use it for in focus or out of focus, focusing image for close and far distance. So that's part of our eye is dynamic, like the pupil is also dynamic. The refractive index is changing. The next part is vitreous chamber that is full of like jelly-like liquid. It has refractive index, but the main function of this part of our eye is to keep the shape of our eye like very spherical or close to spherical, keep the lens at right position, keep the retina at the right position, and don't scatter the light, because the light is passing through this chamber and it shouldn't be any particle that can scatter the light. And the last one, which we can say is the CCD of our eye, is a retina that has a lot of nerves. The nerves send the signal to the brain, and you can see the image. The perception of those signals and chemical and electrical signals is the image that comes to our brain. But we should be very careful, because the image that projects on retina is not exactly that we are seeing, because there is a lot of parts that you have veins, you have blind spot, but our brain make a perception of that final image. And we have fovea, that a very small spot compared to the whole size of retina, that has a lot of nerves, and you can see the best sharp image at that point. When it says your eye is 20 out of 20, they're talking about that part of the eye that you can produce a very, very sharp image, that we use it for example, some kind of activities that you need to see things very well, like reading a book or when you're driving. The image is on fovea. So, the first part, cornea. It has two surfaces. The first surface, we call it anterior cornea, this surface. The second one, we call it posterior cornea, the second surface. When you look at it, it looks a very spherical surface, but in reality, it is a aspheric surface. So it means that it's not like complete spherical. It has like ellipsoid shape. So as I said, it's an aspheric surface, and the mathematical equation that we use for explain this aspheric surface, followed by this equation. And in order to define a aspheric surface in a very simple way, when it is completely symmetrical around Z, is to just define two parameters. Each one of them is Q, the other one is R. Q is a parameter that we call it asphericity. So when Q is smaller than 1, the shape is something like this, hyperboloid. When it is exactly -1, it's paraboloid. And when it is between -1 to 0, it's ellipsoid. But you can see the effective diameter is bigger than the spherical surface. And when it is Q, it's obvious, this is sphere. And when it is positive, it's again ellipsoid, but you can see it, it's inside of that bigger sphere. So here I'm showing some numbers for the anterior corneal radius and asphericity. You can see, for example, for this paper, this is the authors of this paper at 1970. They collected those data for a lot of patients. You can see for 350. This one is 8, it's not that many. 1,000 patient, 176, 220. But you can see after getting average of all those patients, those numbers are pretty close. So it looks like we have a good set of data for that radius. And for asphericity also, those numbers are pretty close, but not like radii of curvature. So this is another paper that I find, for anterior and posterior corneal radii of the same eye, not right and left. The ratio is also, you can see, constant, and those are the number of the eyes. And you can see we have two surfaces. So one of the papers that is very well known, and I see that a lot of people refer to that paper, is a paper from Leo and Brannan, 1997, and they use R1 7.75, the radius of curvature of the first surface, and Q1 that they explain it as asphericity is negative 0.18, R2 6.4, and Q2 negative 0.6, and the thickness of this part of our eye cornea is only 0.5 millimeter or 550 microns. So it's very thin layer, and the diameter of the whole, actually it's not that size, very smaller, is 5 millimeter. And those are all geometrical features of cornea. So we need to have for this part, this thin layer, this thin part in between two surfaces, we need to have the refractive index. And in another video, I completely explain what is the Abbe number. So, but in Zemax, you cannot find any glass from catalog, so you have to put two numbers. One is the refractive index, the other one is the Abbe number. Abbe number is a representative of refractive index is known, the velocity of light inside of the material. But the Abbe number is the representative of the dispersion of the material. When the Abbe number is big, means the dispersion is low. So, but you can see those numbers are pretty close to water, because that makes sense. Most of our body is made from water, and this is transparent part of our body, so it should be pretty close to water. But those are two numbers. You can watch the video that I'm gonna put it in this description that explains Abbe number and refractive index for when you want to model it in Zemax, or generally when you want to define a material optically. You cannot just define it by one number. Your one number is for just one wavelength, but when you put the Abbe number, you're talking about change of refractive index versus wavelength. And our eye can see a big spectrum from 400 to around 700 red to blue. So the refractive index is not constant for that range. So that Abbe number is a representative of that kind of change that cause chromatic aberration in our eyes. So those are some kind of biological thing that looks like not very relevant, but because I like it, I wanted to talk about it. The healthy cornea is lack of any blood vessels, which makes sense because it should be transparent. So in order to receive oxygen, to actually send oxygen to that part, our body use oxygen that dissolve in our tears and that diffuse across cornea to maintain its health. Uh, Sensitivity of the cornea. Cornea is the most sensitive tissue in our body. Research shows that the density of pain receptors in cornea is very high. It's about 300 to 600 times greater than the average of pain receptors in our skin. And the dental pulp has a lot of all the people are very familiar with that kind of pain, but even cornea has 20 to 40 times greater than that pulp pain receptor. So any injury to the cornea can cause a huge pain because of we have a lot of pain receptors in that area. So the second, I mean, the next part of our body is anterior chamber. As you can guess from the name, it should be like a chamber, and the liquid inside is like a jelly type liquid. The name of that liquid is aqueous humor. And again, the refractive is pretty close to water. And we have these two numbers for actually modeling this part of our eye. It's a completely transparent fluid like blood plasma with very low level of proteins, very low uh, dispersion. I mean, not dispersion, scattering, and it maintains the pressure and globe inflation. Hydrostatically, pressure maintains the spherical shape of the eye. So that has a very important function that keeps the cornea in a very spherical, which is actually, as I mentioned, is aspheric, but it keeps the shape and it supplies nutrition, amino acids to the two surfaces that is connected, and potential to transport an antioxidant. And another important function of this part of the eye is that it doesn't let any dust or thin pollen pathogens goes to the eye. So it acts like a defensive mechanism for the eye. And as I mentioned, the refractive index is pretty close to the water, and it prevents eye from dryness because it has a lot of moisture inside. And I think the liquid in between is changing after, I don't know, a while, but it's not a constant thing that you have it from time that you're born, but there is some kind of some liquid goes there, some liquid get out, so it's not a uh, constant water that remains there forever. So the next part of our eye, which is optically very important, is the pupil, which is this side of our eye, which like as it acts like a pinhole, or it's better to say iris because the diameter of this side is changing. And in Zmax or any optical model, it acts like a stop of your system. So that's the main limitation of the field of view. So that part of our eye is not like a static part. It's dynamic. It depends on the humidity. It depends on the intensity of the light, your feeling. Even when you use some kind of drug, it gets really big. So uh, anatomically, the pupil is positioned directly in front of the crystalline lens. So we have pupil, we have crystalline lens, and I'm gonna explain it after this slide. So it is not exactly centered with respect to the eye. So what does it mean? First, I need to explain the coordinate of our eye. I mean, this is something that they use in literature. So our nose, we call it this area between those two eyes, we call it nasal area, and the two sides, we call it temporal. And this, the aperture or the stop of our optical system is slightly offset from the center, close to the nasal point. So we should look at, when we think about it, this is y direction, this is z direction, and in x direction for right eye and left eye, negative and positive, is offset by 0.5 millimeter or 500 micron. This is based on one model that I explained. That's a very well known model, 1997, Brandon and Leo. The normal pupil size in adults varies between 2 to 4 diameter in bright light, because when we are in bright light situation, our pupil size gets smaller because we don't want to get a lot of light and damage retina. And in dark areas, when there is not that much of photons, the pupil size get bigger, 4 to 8 millimeter, and the diameter, based on the model that I explained, he said 2.5 based on Liu and Brandon model. And as I say, in low light 4 to 8, bright light 2 to 4 for adult size pupil. And the material, because it's not empty, it's not just like an empty hole, nothing in there. So there is material inside of even that stuff. The material, as always, is pretty close to water. We have 1.336 for the refractive index and Abbe number of 50.23. So the human pupils are typically, it should be circular. There is a lot of papers that they can actually, they said no, it's not completely circular oval shape, but we consider as a very close to completely circular shape. But for some other animals like cat and reptiles is completely vertical. And I read a paper about it. The reason for that is that for those kind of like cats, because they want to hunt the other animals, when the pupil is vertically, they can get a better estimation of the distance to the thing that they want to eat or they want to hunt. And for goats and sheep is horizontally, and the reason is completely opposite of like cat and reptiles, because they want to see a big field of view in horizontal direction, because during the night something can attack them, so they need to see a very big field of view. And for some catfish it has annular shape, that I didn't do that much of research about this part. So the next part of our eye after stop is crystalline lens that has two surfaces: anterior surface, the first surface; posterior surface, the second surface. And the refractive index of this part of our eye is not constant, and I have another video completely explain the mathematical equation, the gradient index of that part of our eye. The main reason that we don't have that much of data about this part of our eye is that, first of all, it's very deep inside. There is not that much of studies compared to corneal shape, because it is outside of our eye. You can even physically measure it, or if you want to optically measure it, because that's the first surface, much easier than if you pass through the cornea and measure the lens. And that lens also has two situations: relaxed, accommodated situation for far and for close and far distance. So there is not that much of results or studies about it. And the results obtained by two main category: in vivo and in vitro. In vivo, vivo is a Latin word for live, for something that is live. So in vivo means by doing some kind of measurement, like optically or even physically, I don't know, you can measure something in a live animal or live human. You can measure it directly. Vitro in Latin means glass. So it means that in experimental or in lab equipment, you can, when for a dead animal or anything, you can take it out and measure it in the lab. So we have two sets of data, live human or animals or the lab. And here is based on, again, Liu and Brannan model, 1997. So you can see for the lens, we have grade A, grade B, which means that the refractive indices gradient is not constant. So the radius of curve, we need two parameters, radius of curvature, first surface and second surface is 12.4, negative 8.1, and we have the thickness for each lens. So we have some kind of imaginary plane between two lenses, and we have a thickness for each of them. On next slide, I'm going to explain it. So I have this table again. That's the first part of the lens in our eye, that the anterior surface. So this surface, we have the radius of curvature of 12.4 and the thickness of 1.59 based on this model. And the second surface, you can see it has a lower radius of curvature, which is negative 8.1, and the thickness is 2.43 millimeter. So in another video, I explained the refractive index. The refractive index is not constant. At the center of this part, we have higher refractive index, and when you go to those two directions, in y and negative y, the refractive index goes lower. C is the same, and we have this equation. You can watch that video to get a better understanding of gradient refractive index, but we have those numbers based on the same model that we can import it into Zemax or any other optical model in order to optically model this system. The next one, as you can guess, is nothing. Vitus chamber that is full of, again, jelly-like liquid that we call it, and this is the part of the eye that we call it vitreous humor. And again, refracting is pretty close to water. We have Abbe number 50.23, and this part, the main function is to support the lens, because we have the lens here. This jelly-like liquid, push it, make the spherical shape of the lens. It maintains the entire chamber or posterior cavity. This side we call it posterior cavity, posterior cavity, and the composition of this fluid is 99% water and contains no cell. Because it makes sense, if you have cell, you have scattering, but this is a part of the eye that light is passing through. We don't want to have scattering. And when you get old, there is a chance that you can see something in when you're looking at the objects, some small particles that goes to that liquid and make a scattering, and that scattering cause some kind of seeing some object that they call it fly something, that that's a kind of disease that because of that water has some kind of particles in it. It's a very pure jelly-like particle of water with no cell. So the next surface is retina, which is like a CD of our optical system, or image plane of our optical system, that the image appears on that retina. So the retina is this part of our eye. I look at a lot of literatures. Most of them, they use negative 12 millimeter for the radius of curvature. One of the main difference between our optical system, our eye, or any other optical system like cameras, is that most of them you have a flat CC, but our eye is curved, which you don't have distortion for our eye, and but we cannot make CC like curved like our eye. So it has layers has different layers with photoreceptors and glial cells, that some of them are for like color photoreceptors, some of them are intensity photoreceptors, and uh, the image that appears on retina is not exactly the image that we see in our brain, because our brain fixed that image. So it's like post processing, because some part of the retina you have a lot of veins, and then you have blind spot, but our brain fill those gaps and give you a nice image. And as I said, fovea is a part of, very small part of a retina, that you have very sharp focus image. And the main function of retina is to capture light and convert it into electrical and chemical signal, send the signal to brain for visual perception. Thank you very much for your attention. Here is the references that I used, most of it from the Optics of Human Eye, that book, and papers, and those are for some photos. So please put some comments for the next video and guide me what you like about it, what you want to learn more about optical system, that can help me to provide more useful videos. Thank you very much.